Assalamualaikum, a very good day to everyone and especially to our lecturer, Madam Fariza Aina. We are from AS1205J and today we are going to discuss about experiment 5, which is gas chromatography. And this is our group members that contain 6 students. So first Adriana Sarah, Mastura Hazira, Rabiatul Maisarah, Idina Hani, Nurin Amirah, and Nur Atika Syairah.
Hi, my name is Nur Atika Syairah binti Nazri. I will present about the introduction of gas chromatography. Gas chromatography is an analytical technique used to separate, identify, and quantify volatile compound in a mixture. In this method, the sample is vaporized and transported by an inert carrier gas through a column containing a stationary phase. Different components of the sample travel through the column at different rates due to variation in their volatility and interaction with the stationary phase, resulting in effecting separation. Gas chromatography is widely used in environmental analysis, pharma- pharmaceutical quality control, food analysis, and forensic investigation because of its high sensitivity, accuracy, and rapid analysis time. The objective of the experiment for gas chromatography is to determine the retention times of N-butanol and 2-propanol. Second is to identify the component present in a standard mixture based on retention time. Third is to identify the component present in unknown sample. And lastly is to determine the effect of temperature and retention time and resolution. Next, for the apparatus we use in this experiment is syringe, beaker, and dropper. For chemical, we use 2-propanol and butanol, standard mixture of 2-propanol and butanol in 1:1 ratio, and lastly we took random sample which is an unknown sample.
Next, we will move to the methodology of the gas chromatography. So, gas chromatography is an analytical technique used to separate, identify, and quantify volatile compounds based on their interaction with a stationary phase and their volatility. First, from an inert carrier gas, such as helium or nitrogen, is supplied from a gas cylinder. The regulator controls the gas pressure and flow rate. The carrier gas acts as a mobile phase, transporting the sample through the gas chromatography system without reacting with it. So, the outer sample introduces a precise and reproductive volume of the sample into the system. This ensures consistency between injection and minimize human error. The samples is usually injected in liquid form. The sample will enter the heated injection point where it rapidly vaporizes. The high temperature ensures complete conversion of the sample into the gas phase. So the vaporized samples is immediately swept into the column by the carrier gas. The vaporized sample travel through the gas chromatography column, which contain the stationary phase, and it is housed inside the temperature control column oven. So different compounds interact differently with the stationary phase based on the polarity, boiling point, and molecular weight. Compounds that interact weakly with the stationary phase move faster, while those with the stronger interaction will move slower. This causes separation of compound, each elevating at a different retention time. As each separated compound exits the column, it passes through the detector. So, the detector converts the presence of the compound into the electrical signal. The signal intensity is proportional to the concentration of each compound. The detector signal processed by the computer producing a chromatogram. So each peak represents a different compound and the retention time help identify compound while the peak area or height is used to quantification. Next, we will move to the component and the function of gas chromatography. First, we have the sample inlet, also known as the injector. Its main function is to introduce the sample into the gas chromatograph. The injector heats the sample so that it's vaporized and is converted into the gas form, which is necessary for gas chromatography analysis. There are several types of injector, which are split, splitless, PTV, or on-column injector. Among these, split and splitless injector are the most commonly used depending on the sample concentration. Next, we have carrier gas system. The carrier gas is an inert gas such as helium, nitrogen, or hydrogen. Its function is to carry the vapor sample through the column. Because the gas is inert, it does not react with the sample. A constant and controlled flow of the carrier gas is very important because it affects the speed of analysis and the quality of separation. Then the column is a long tube that contains a stationary phase. It is where the actual separation of compound takes place. Different compounds in the sample interact differently with the stationary phase, causing them to travel at different speed. As a result, the compounds are separate and exit the column at different times. So column can be packed or capillary column, and they are placed inside the temperature control oven to ensure consistent and accurate separation. Next is the gas flow regulator. Its main function is to control the pressure and flow rate of the carrier gas. It ensures the carrier gas is delivered consistently and steadily, which is important for obtaining accurate and reducible result. If the gas flow is not properly regulated, the separation of compound may be poor or inconsistent. Next is the oven, which contains the column. The column is responsible for maintaining and controlling the column temperature. So it can operate either in isothermal mode, where the temperature is constant, or in program temperature mode, where the temperature increases gradually during analysis. So proper temperature control is crucial for efficient separation, especially for compound and with different boiling point. After the compound pass through the column, they will enter the detector. So the detector's function is to identify and measure the compound existing in the column. So it produces the electrical signal proportional to the concentration of each compound. So different detector can be used depending on the type of compound that being analyzed. And finally, the computer and data analysis system, it record and process the signal produced by the detector. So it display the result in the form of chromatogram, which show peak corresponding to the different compound. This system allows for qualitative and quantitative analysis, as well as data storage, calibration, and report generation.
Next, I will explain about results using GCMS. In this experiment, we have 3 compounds, which is N-butanol, 2-propanol, and the mixture of N-butanol and 2-propanol. For the first compound, which is N-butanol, by looking at the graph, we can see that we have one peak with 0.217 cm width and 6.911 second retention time. For the second compound, which is 2-propanol, we got one peak with 0.109 cm width and 5.783 second retention time. Lastly, the mixture of N-butanol and 2-propanol. By looking at the graph, we have 2 peaks and we need to identify which one the N-butanol and 2-propanol. So for the first peak with 0.1114 cm width and 5.78 second retention time, it is for 2-propanol compound. And for the second peak with 0.155 cm width and
6.895 per second retention time. It is the N-butanol compound.
Next, I will present the questions of this experiment. First question is state the types of compounds which are suitable for analysis using GC. There are two types of compounds that are suitable for analysis using gas chromatography, which are gas solid chromatography and gas liquid chromatography. For GSC, the mobile phase is gas and the stationary phase is solid, while GSC, the mobile phase is gas and stationary phase is liquid. Next question is why is FD suitable detector for this analysis? Flame ionization detector or FID is suitable for gas chromatography analysis because it can detect a wide range of hydrocarbons. A FID contains a collector plate with a hydrogen or a flame. The flame which breaks down organic molecules and creates ions receive the effluent from the GC column. An electrical signal is created when the ions are collected on a biased electrode. The last question is list two factors which can increase the efficiency of a GC column. Two factors which can increase the efficiency of a GC column are the dimension of the column, where the separation can be improved by increasing the length of the column. Decreasing the particle size of the matrix also can improve the separation in the GC column.
So our next part is discussion. In this discussion, we know that retention time can measure the time taken for a solid to pass through a chromatography column, where they calculate from the time between sample injection and detection. Next is factors that affect the retention time, where factors affecting retention time is carrier gas flow rate, oven and column temperature conditions, column length, and column foundation or degradation. For chromatograms result, all chromatograms produce distinct peaks with varying results. There is 4 sample analyzed, which is 2-propanol and butanol, standard mixture of 2-propanol and butanol, and the last one is unknown sample. And our fourth point is experimental condition, where our temperatures use 17 Celsius, 100 Celsius, 140 Celsius, and the last one is 180 Celsius. For chromatogram observation for 70 Celsius, there is two mixture, which is the first one is standard mixture chromatogram show there is two peaks, which is 5.772 min and 6.882 min, and individual chromatograms of 2-propanol and N-butanol show one peak, which is 6.916 min. 6 is effect of temperature on retention time, where as we can see, effect of temperature on retention time can increasing temperature result in shorter retention times, and different oven temperature produce different chromatogram patterns and peaks. For chromatograms interpretation, standard mixture at 70 show longer retention time have two distinct peaks, where it presence of two peaks indicates two different compounds in standard mixture. So the last one is compound identification, where we can make comparison with individual chromatograms of 2-propanol and N-butanol, where we found that it was similar retention times, confirmed that the standard mixture contained both 2-propanol and N-butanol.
Lastly, conclusion. To conclude, this experiment was a success as all objectives were achieved. This experiment successfully determined the retention times of N-butanol and 2-propanol, which were found to be 6.992 minutes and 5.77 minutes respectively. The component present in the mixture were identified based on their retention time. In addition, the effect of column temperature on retention times and dissolution was studied. Increasing the temperature resulted in shorter retention times due to faster elutions of analytes, while lower temperature generally provide better resolution.